of a number of platforms all on the market, ranging from looking at the 3D genome to functionalized cell imaging. And we also have a number of spatial biology platforms, our COSMIC system, GeoMx, and our CellScape, and our nCounter, which is a bulk profiling system. So in Bruker Spatial Biology, Bruker has kind of accumulated a number of platforms together. The two I really want to talk about today are our GeoMx and COSMIC systems, but I'll just mention that we have split our platforms into a number of kind of topics. The first is cell biology, which is our nCounter system. It's very robust. It's done on bulk RNA samples. You can profile up to 800 RNA targets in one tube, so multiplex. And then we have our Beacon. There's an extremely large one in our Beacon Discovery. And these are really for functional profiling of live cells, so used a lot in CAR T therapy and cell line development. I'm going to be focusing on spatial biology, and in particular our GeoMx and our COSMIC systems. Both of these systems are multi-omic profilers, meaning we are profiling RNA as well as protein in a spatial context. That means we're not dissociating cells, we're taking slices of FFPE material or fresh frozen material, and then we are profiling RNA and proteins in two different manners. So with our GeoMx, we're really focusing in on structures and collections of cells, whereas our COSMICs, we are looking at single cell and subcellular resolution of those molecules. So just to take one step back, why are we doing spatial biology? What is spatial biology? And I want to start this with a very simple example, which is two different tumor samples from patients that were included in a trial or in a study to response to a drug. And when these two tumor samples were profiled with bulk expression, so RNA sequencing, they gave a very similar tumor inflammation score, meaning there were a large number of immune cells present. However, one tumor, the one on the right, responded, or the person with that tumor, and the one on the left didn't. If you just looked at the tumor inflammation score, you would wonder, why is that? But if we look spatially, so if we look in this tissue, profiled in green is PanCK, so tumor cells, and in this magenta, it's marked for CD45, so the immune cell marker. And you can see in the tumor on the left, these immune cells are completely excluded from the tumor area. Whereas we look in the immune cells on the right, we can see they're infiltrated in this tumor. So this is a question that if we didn't have spatial context and we didn't have a way to explore these cells in a spatial context, we would have a hard time getting to an answer. However, this was done with the GeoMx system where we're able to use the protein stain, so use the PanCK or the CD45, to extract whole transcriptome information non-destructively from our immune cells that were infiltrated and non-infiltrated. And what we found was actually a very distinct cluster of genes expressed in the TME, so the tumor microenvironment within the infiltrated tumor. So I hope this gives us a kind of a sense of why we're doing spatial biology to begin with. So that study was done on the GeoMx, and the GeoMx system really has unmatched spatial multi-omics plex. On the market, we have assays that look at whole transcriptome for mouse and human. We also have two cancer-focused assays for human and for canine. And you can also profile over 1,200 proteins at the same time on the sample. We're using biology to really drive where we're asking questions. So we're not profiling non-discriminately. With the GeoMx, you're actually choosing the area that you're interested in, so choose a region. And then you can focus that in further using protein stain that you've done on your tissue to really just pull context from a collection of cells or a structure. So I want to just start with an example here. This is another tumor used a lot in oncology research. So this is another tumor. And what we've done is we actually have our tumor here with multiple different tumor regions. And there's a stain, it looks nice, you have PanCK, you have CD45, but that's not the interesting part of what we're pulling out in our system. We have stained this tissue with both our RNA reagents and our protein reagents. And I apologize, it's a little bit small. But what we are doing is we are harnessing UV light to help us focus in on regions. And then we can expose the tissue to the UV light where we have hybridized our RNA reagents. There's this photocleavable linker. And then the only thing we're taking off our sample are these small little DNA barcodes. They're 26 base pairs, float into solution. We can pick them up with a microcapillary zipper, move them into a 96-well plate, and sequence them on an Illumina sequencer. And we use the same trick for proteins. So we have our antibodies that are using a photocleavable linker to, again, have a small DNA barcode attached to them so that we can have a large number of antibodies on our tissue without thinking about steric intrinsic fluorophores, where if we turn on UV to a specific set of cells, we release these tags. So again, here's just an example where in green we have pan-CK, and we're looking at one of our tumor regions, and we have pan-CK in a non-tumor region below. These have been annotated using an H&E, so you can look at an H&E ahead of time and then choose the areas within your tumor you're interested in. And what the system does is it can see your protein stain, and it uses it to create these masks. So here you can see in gray is one mask and this yellow color is another. The system will then turn on the UV to only the area where it has decided to mask, and it does this with digital micro mirrors. So we have 1 million little mirrors, and they're on a tiny little chip about the size of your thumbnail that will focus the UV light onto the specific set of cells, which will then release these tags, and you can profile your sample. So you may think, okay, well, why are we going to profile RNA and why are we going to profile protein? And another thing I didn't mention yet is in our 1,200 proteins in our panel, we also have a large number of post-translational modifications. So what does this tell us? When we're thinking about samples in biology, there are a large number of different layers that all integrate and all tell us something. If we think about RNA, it tells us what's the potential of the cells I'm looking at. What is there? What RNA is there? If I look at protein, it says, okay, well, has this RNA gone into protein? So is the machinery present? Is there the capacity within those cells to perform what I see in my RNA? And lastly, with post-translational modifications, you get the idea of activity. What is actually active right now? So by combining multi-omics in this study, we can really distinguish transcriptional priming from what's functionally happening in these samples. So I want to give just one example for the sake of time with this same tumor cell where I have my normal and my different tumor regions. So these green blocks are denoting the normal tissue, and here we have our tumor regions that we've collected. And if we just look at this heat map very roughly, the top line is TP53 RNA. And we see that it is elevated in tumor. We expect that, right? It's a canonical pathway. We expect it to be elevated in tumor samples. And if we look at our P53 as well as the acetylation in phospho forms of P53, we also see that they're elevated in the tumor as well. So we know that the DNA damage pathway is activated, but we also can see, okay, which pathway is actually activated within DNA damage? Is it single-strand repair or is it double-strand repair? And there's actually quite an easy way to tell the difference between this, and that's by looking at the ATM or the ATR. And in our protein panel, we actually have a phosphorylated ATR antibody or a phosphorylated ATM. And so what we see is that in our tumor cells, we actually have an upregulation of phosphorylated ATR, whereas we see higher phosphorylated ATM in our normal tissue. So then we can actually say, within this tissue, okay, we had the machinery for the TP53 pathway, we had our proteins, but what we see is that our DNA damage response within our tumor regions is driven by single-stranded DNA repair pathway and phosphorylated ATR. So really kind of putting everything together within this context. This system is able to profile 4 slides at a time. You're also able to just focus in on the areas of biology that you care about, so that helps streamline your analysis and also help you get to an answer faster. When we think about spatial biology, there are multiple levels of spatial resolution, though, that are required. So what I was just looking at with the geomics is I'm looking at tissue structures or groups of cells. We need anywhere between 50 and 100 cells to get good profiling for RNA. You only need a single cell for protein just because they're much more abundant. That will answer a lot of questions. However, there are questions that are really focused more on cell-cell interactions and finding a rare cell type or a specific kind of T cell within a tumor. To answer those questions, you need a platform that can do single-cell resolution as well as sub-cell resolution. And for that, we have our CosMx platform, where we're looking at single-cell resolution. So the CosMx, it's again a multi-omics platform where we're looking at mRNA within our tissue non-destructively up to whole transcriptome. And you can also profile up to 72 proteins independently or at the same time on the same slide. I will also just quickly mention that the first CosMx in Romania will be installed within the next few weeks at the Victor Bravish Research Institute, which is just down the road. So that's very exciting that this platform will be available in the neighborhood. So with this platform, we really have best-in-class sensitivity. We're taking 8 Z-stacks through your 5 micron FFPE piece of material or fresh frozen organoids. Works on basically anything since it works on FFPE. We're looking at whole transcriptome imaging at a subcellular level. We have same cell multi-omics, and I'm very excited to announce that we actually now have 3D cell segmentation built into the platform. It was just released a few weeks ago and is now being rolled out into the field as well. When we think about the CosMx system and what we're doing, we're not choosing an area within our tissue. We are putting down fields of view on our tissue, meaning that we can tile a large piece of tissue. This is a lymph node. You can also put a TMA on a slide and profile those different TMA cores. It's a 0.5 micron by 0.5 micron square that you plop down, so this one's been tiled by a large number of them. Within that, we are then profiling every single cell within the area you selected. So within this lymph node here, I'm showing single cells. So each of these single dots represents a different cell, so with cell typing. But we go even farther, and we have our 8 Z-stacks through the tissue with 50 nanometers resolution to actually pinpoint where transcripts were with an XYZ location. So these dots are cells, these dots are transcripts that we're able to look at. It's a large number of cells that you're pulling from every experiment, so quite a bit of data that you get. The way that we're doing this and the way that we're able to overcome optical crowding is by the chemistry that we are using to read out this assay. You have your RNA that's within your tissue section, and we're hybridizing a set of probes onto your RNA, so up to whole transcriptome, which is over 19,000 probes. With the geomics, we're using a photocleavable linker. That doesn't work with CosMx because we want to know exactly where a transcript is within space. So we've taken that same chemistry and we've modified it by creating reporters that we flow in different rounds of cycling that hybridize to a landing domain or readout domain. So we're able to read different spots in space, and essentially we're creating an optical barcode that we will then decode. And I'll go into that a little bit more. I think it's useful to just kind of show you what this looks like on the instrument. So this is actually a tonsil sample that I ran in the lab. The tissue is on normal histological slides, so just regular glass slides, nothing special, nothing coated. We are doing a day of slide prep where we are, antigen retrieval, deparaffинizing, and then an overnight hybridization with our essentially modified ISH probes. You can also add your antibodies, which are going to be read out in a similar manner. These are not antibodies that are fluorescently labeled. They have, again, readout domains, and we are flowing in reporters to read them out. So the first thing that happens on the system is you're going to see the first round of imaging where we've looked at a set of what we call morphology markers on the tissue, which are really just there to help you put down your fields of view. This is low resolution, quick scan that we do. You then are going to put down your fields of view. So there are different squares that you can put down, tile them, make a grid, put them randomly, whatever you want to do. And then the system is going to start cycling, and it's going to take a high resolution image of that same area where you put down your fields of view. So now we're again doing our morphology readout, but we're taking 8 Z-stacks and doing a projection of them. That's what you'll first see. Or you can later use them to do 3D segmentation of your tissue. On the instrument, there's also segmentation that is done of your cells, because if you find a transcript in space, it doesn't mean anything unless you can place it within a cell boundary. So this is something that we've really spent a lot of time working on, is doing this cell segmentation. So I want to talk about this just a little bit more. We have had the best-in-class segmentation from the beginning because we've always felt it was important to include a number of morphology markers to really define the boundaries of cells. We have been taking these 8 Z-stacks, projecting them in 2D, and doing cell segmentation, which is kind of the norm. But now you're able to take these 8 Z-stacks and you're able to redo your cell segmentation. You can think about this, if you take a 5-micron slice, you hope it's single cell, but in reality there's always going to be some cells that are overlapping that you've caught. So now if you're able to do 3D segmentation, you're able to really assign transcripts to the correct cell and make sure that we're not getting bleed over between different cells that just happen to be on top of each other. We've also worked with a number of ABI models to help with neurospecific and also muscle specific, as these tend to be quite difficult to segment. To just show a few images of how we are leveraging our AI model, we have actually modified algorithms that we have now AI trained on a large number of samples, Brooker-generated samples, to be able to have really great cell segmentation. And this can also be further fine-tuned in our Atomics cloud platform, where you can then change the parameters to be more inclusive, less exclusive of certain cell areas. So once we have got our segmentation, we are actually doing reporter cycling and in situ imaging. So depending on the plex of the assay that you're looking at, this is going to be a different number of cycles. What we're doing is we have already prepped our sample where we have our probes hybridized to our RNA within our tissue. We then flow in a set of reporters, so 4 different reporters per round. They will bind or hybridize to our readout domain. We wash off the extra ones, and then we're actually taking a picture. I'm skipping forward. We wash them off, and we do this depending on the plex of your assay a large number of times. And the end, what you're generating is, here we have 2 examples, very simple, of 2 genes. This is a 1000-plex assay where we're basically creating a barcode. We always have 4 cycles that will give a signal. There's 4 landing domains, and the rest is dark. And that is also helping with optical crowding, so we can really distinguish all of these different transcripts in space. So thinking about this technology, it came on the market at the end of 2022, and we have gone from having a very small 100-plex, 1000-plex assay now to whole transcriptome. And we actually have publications, peer-reviewed publications on the market, or out in, I guess, in the wild, we would say, where we're looking at, aggressive B-cell lymphomas. There's been a CRISPR screening that was done at Merck, where they're doing using the cosmics to really help see which guide RNAs got in. And there's also different tracing trajectory of cancer progression in space. So these are all available to look at. What I want to focus on now is I want to just talk about a data set that also is in the public domain that we generated, which is a multi-omics breast cancer data set, where we have whole transcriptome as well as the 64-plex immune oncology antibody panel that we looked onto. And you can actually download this data and look at it yourself. I don't have a lot of time today, so I just want to briefly talk about a couple things within the sample. And the first one is really a paradigm shift in how we're thinking. A lot of spatial biology is using small targeted panels. When we do that, we're able to cell type. When we do cell type, we start thinking about, okay, if I have these cells here, this is likely what's going on. However, when you have whole transcriptome data, you no longer have to guess what's going on. You can see what's going on. So there's no longer this inferring interactions based on cell typing alone, but now you really have the direct biology measurements across the tissue. So I'm taking the same, this is a breast cancer sample where I have a primary tumor and an invasive front. And instead of just saying, okay, I think this is what's going on, what I can actually do is I can go through over 2,500 different pathways within that sample, because I have the genes there that I've read their expression out for, and I can say, okay, well, what's going on with a number of different pathways within my tumor? So now I'm projecting them. So we're looking at, these are all pathways that are regulated by hypoxia. So I can look at, what is my MAPK expression across the different areas of the tumor. TNF alpha, I then can see, hey, this is really elevated within my primary tumor and not as highly elevated within this invasive front, except at kind of this leading edge. And the same for JAK-STAT. So, this is only 4 pathways that I look at, but you can go through and look at all of these. So instead of guessing, you just go take a look. Spatial biology, it's there in your tissue, what's going on. Oh, I'm missing a large number of slides. Okay, that's okay. I'll just go back really quickly to talk about this primary tumor area as well as this invasive front. So within this invasive front, we can actually see two different domains. So what we're looking at is we are looking at cells within the area, so I have cell typing, and we can start breaking those into what we call niches or neighborhoods. So what collections of cells are always near each other? If I look into my invasive domain here, I actually have two different kind of domains. We're naming them by the color, so we're calling it this, it's called tan, but I think it's a little bit orangish. Everybody's eyes are slightly different. And we have our light blue area also within this invasive front. What we see is that if we look in this kind of orangish tan color domain, we have very proliferative pathways that are activated that are, showing that these cells are very active, that they're invasive. And if we look at the blue area, we say, okay, well, this is kind of the wasteland after these proliferative cells have gone through. There's a lot of stress response, there's a lot of nutrient deprivation pathways that are going on. So I'm missing the slide, but then what we actually can do is now we're looking, okay, these are the neighborhoods, but if I focus in on this kind of area up top, I can then focus in on the cell types and say, okay, well, what cell types are actually present here? And what we found is there was a large number of CD8 T cells that were activated at this front. And if we compared CD8 T cells from the edge of within this kind of proliferative domain versus the internal domain, you can actually start getting very unique profiles to different they have the same kind of classification of T cell, CD8 positive, but they're actually the expression that they're expressing is very different. So with this, because we're profiling not only areas, but every single cell within here, we can start making more comparisons and pulling out what is different between a T cell that's able to be in this kind of invasive area and that's on the leading front. Okay, so that was all looking at just RNA and information that we're getting from RNA. With genomics, I told you, yeah, protein's also important. Well, in cosmics and in single cell, we also think protein is important as well. So what we can do here with cosmics as well is you can do same cell multiomics, where we're combining any of our RNA panels with our 64-plex immune oncology panel that can be further customized. What do we get when we combine cosmic same cell multiomics? We actually can show RNA-protein relationships at a very high plex. We can orthogonally validate biomarkers on the same slide. So if you find an RNA that's up, you can already see the protein if it happens to be in the panel as well. And then you can also incorporate tissue architecture into your spatial analysis. I want to just give an example here. This is a non-small cell lung cancer sample. So again, another sample where we have looked at both RNA as well as protein on the same slide. And here what we're looking at in the kind of more blue images is the RNA pathway, so MHC class 2 antigen presentation and interferon signaling on the right. In our protein panel, we also have HLA-DR protein, which is the active molecule within this MHC class. So you can see, okay, where I have increased MHC antigen presentation, I also have increased HLA-DR protein. So this is kind of showing, okay, RNA and protein matches. We all know that's not always the case though, right? There are times where it's correlative, anti-correlative, where it doesn't match at all. And that's actually what you see here with the Vistra protein. This is not a novel interaction. It's known that when the Vistra protein is present, it's actually inhibiting interferon signaling. So you see where we have a high level of Vistra protein, we don't have interferon signaling, and where we have a lack of Vistra protein, we have a very high level of interferon signaling. So again, at the same time, we can just look at this all in space. And I will now just focus in on one little area here within the same experiment where I have two different tumor beds. So here I have one here in blue, these are both tumor cells, and in the middle I have a large number of immune cells, immune infiltrates. This is our protein image, just from looking at some of the proteins within the cells. And here is our RNA image, so looking at our whole transcriptome. And something I want to point out is that there looks like this area here where there's no transcripts and there's no cells. I have a hole in my tissue, it looks like. However, if I go through my panel of proteins, so this is just a number of proteins where we're distinguishing our tumor and our immune infiltrates. If I, for instance, put on fibronectin from this panel, I can see that there was a large amount of fibronectin here, small amount down here, but you see at top, it's really creating this extracellular matrix. It's blocking out the immune cells from getting into that tumor nest. I can do the same thing for smooth muscle actin. Sorry, too fast. So down here, I see that there is a large amount of SMA, so smooth muscle actin, that on this tumor bed is creating this kind of block. So as we start piecing together our protein and our RNA, we can get a lot more answers, because if you have the correct immune cells that you think are the T cell that's going to kill your tumor, if it's completely blocked out by a protein barrier, you can't get in. So now you have to think about, okay, well, what can we do to help those cells get through this protein barrier? We can also kind of think of new ways that we can really attack cancer biology in this case. I'm missing slides I mentioned, sorry. But we do see SMA protein in the same area. However, what you'll often see is that RNA is kind of sparse, because you have your protein machinery that instead of making one protein for one RNA, there's a lot of protein molecules that are made for one RNA. And that's really what we see here with SMA. If we would have looked at the SMA in these cells, RNA, you would see it there, but you wouldn't see this major blockade that we're seeing. You can go farther and you can start building up even more layers of information. A couple are just projected on here. I'm not going to go into all of them, where we're pulling up different information from our protein and our RNA to really help us with insights. So there's often the question that we get, why are we using such a high-plex panel? What do we need? If I'm just going to look at 64 proteins, why am I doing it? So I just want to quickly highlight two studies, one that was done with the WTX panel and one that was done with our 6K panel, where we were looking in IBD colon samples, or the researcher was looking at IBD colon samples, and they found olfactory receptors being activated in these IBD patients. This is nothing that basically IBD researchers would have been looking at to begin with, but because that target was in the panel, it came up, and they could actually go back and they found one review that was published in 2021 where somebody had mentioned, I think this may be happening, and then now they actually can confirm it. The same thing, this is a PDAC sample where we were looking in the tumor bed, and they actually found that there were nerve cells in the tumor microenvironment that were affecting the tumor immune interaction. So when you're taking a panel that's really large, you start finding biology that you wouldn't necessarily find if you took a targeted approach. If you take a targeted approach, it's not going to be there, you're going to miss it. When we're looking at all this data, it's a lot of data. And we actually have some ways to help researchers look at this. We are using our Atomics cloud platform. Because it's large data, the images are all uploaded into Atomics. You could do 3D resegmentation. And we have two different ways that you can look at your data, which is, you make a study and you do all of your protein, your analysis pathways yourself. But we've also released something recently called Spatial Discovery. And what we're looking at here is a colon whole transcriptome sample where we have done a spatial discovery study, and then we go and we talk to, in this case, Gemini Pro, and we say, tell me a few things that are interesting in my sample. You load your spatial data from a small package that you download. It takes about 30 seconds, it's very quick. And then it tells you, hey, why don't you look at smooth muscles in a specific cluster, and there's two different kinds of cells. I think it's telling you that it believes they're smooth muscle cells. And in this case, it's saying, okay, look at this smooth muscle area, and then look at a different cluster that we think are microfibroblasts that are invading this smooth muscle layer in the columns, in the colon. So we're trying to help researchers get to answers faster as well through our data analysis software and our AI models, because we know it's a lot and we know it's difficult, and we want any researcher to be able to look at their data, and then you can go back and spatially turn on all your different pipelines where after you've gotten some help from Gemini or Copilot or whatever AI tool you're allowed to use. This colon is actually also publicly available to download, so you can go download this. And another thing that we have developed is something called our Cosmics Analysis Scratch Space. These are blog posts where we give you a data set and we walk you through all of the code that you need to use in R, or some of them are in Python, to be able to analyze data. So we're trying to, again, make these tools accessible to everyone, where we know that when you generate data and when you generate large data, the bottleneck is getting to the finish line, because you need bioinformaticians, you need people to kind of really take a long time to look at it. And we're trying to kind of reduce those barriers within spatial biology. So I encourage you to take a look at that if that's interesting to you. I've also shown a large number of just single tissue sections. So I just want to mention that yes, it's very cool to look at a big section, but this can be applied to larger cohorts. So this is just an example here coming out of Marutha Kingslaga's lab, where it's actually a multi-site study where they've looked at over 500 non-small cell lung cancer patients to look at, what's going on between responders and non-responder programs, pathway modeling, and you can really find, in this case, they found unexpected immune suppressive programs that wouldn't have been able to have been discovered with a targeted panel. So you don't have to think small, you can also dream big within spatial biology. With that, I am going to end my talk with just saying, within Bruker Spatial Biology, we have a large number of these instruments, and they're all, playing together, and they're all, not interchangeable, but they're supportive, and they're not necessarily, the fact that you can use one doesn't mean you can't use the other, right? They're all helping, and the data is feeding into each other. So with that, I'll stop. Thank you for your attention.
Amanda, very interesting insights in the portfolio of Bruker. By the way, these seem like very expensive instruments, right?
Well, there's one coming in down the street, right? So
yeah, you're right. So, any questions for Amanda?
Yeah.
Is this
protocol? Very short, because we have one thing.
Yes, a short question.
Lunch exactly in half an hour should end.
Yeah.
So please be...
Just a short question, if this protocol is applicable also to cultured cells?
Yeah, so there's actually a protocol published by Holger Heinz group called STAMP, where they have looked at culture cells on slides. So I would look that up.
Yeah.
All right, and I'll be here for a few more minutes, so I know everybody wants to eat. It's late, we're behind, so thank you guys for your attention.
Let's thank all the speakers once again.